आज जो आपको मेंटर पढ़ाने वाले हैं वो है इंसेक्ट फिजियोलॉजी.
What is this Rodnius prolixus? This spiralous site is important. Here in some insects there is a modification. It forms the diverticulum midgut, which is also called as mesenteron or ventriculus. So gizzard is proventriculus before the midgut. So it is proventriculus and midgut is ventriculus. So here we cannot see any cuticular lining. So important structures in midgut. So in foregut also we have divided into four. Again in midgut also we will divide it into rectal pads play a role in the water absorption and absorption of essential ions and amino acids. And all insects require ascorbic acid.
डियर स्टूडेंट्स, अगर आप ICAR PG 2027 की प्रिपरेशन कर रहे हैं और आपने अभी दिमाग बना लिया है कि मैं एंटोमोलॉजी और नेमेटोलॉजी से प्रिपरेशन करना चाहता हूं, तो आप लोग एक जेन्युइन प्लेटफॉर्म की सर्च कर रहे होंगे कि मैं कहां से अपनी तैयारी शुरू कर सकता हूं. कौन सा प्लेटफॉर्म है जो आपको बेस्ट गाइडेंस, बेस्ट मेंटरशिप और बेस्ट क्वालिटी कंटेंट दे सकता है, और आप इसी को ढूंढते हुए IARI वाला के इस प्लेटफॉर्म पे आए हो. आज जो आपको मेंटर पढ़ाने वाले हैं वो है इंसेक्ट फिजियोलॉजी. इस पर्टिकुलर टॉपिक में अगर आप जीरो नॉलेज रखते हो इस टॉपिक से या एक्सट्रीम 100% नॉलेज रखते हो, तब भी आपको वीडियो देखना चाहिए. आप देख पाएंगे कि हमारे जो मेंटर है वो आपको कैसा पढ़ा रहे हैं, उनका कॉन्सेप्ट, आपका कॉन्सेप्ट वो कैसे इंप्रूव कर रहे हैं और आपको कैसे वो सिखा रहे हैं. अगर आप पूरे साल उनसे पढ़ना चाहते हो तब जाके आप उस बैच में एनरोल करो. आज जो आपको पढ़ाने वाले हैं वो 2025 में ऑल इंडिया रैंक 1 UR कैटेगरी से एंटोमोलॉजी और नेमेटोलॉजी से खुद रैंकर हैं, और वो आपकी आज क्लास लेंगे. ये जो क्लास होगी आप लोगों के लिए एक डेमो वीडियो होगा कि हमारे मेंटर्स कैसे पढ़ाने वाले हैं. सो आगे जाने से पहले, विदाउट वेस्टिंग योर टाइम, अगर आप सीरियसली प्रिपेयर करना चाहते हैं ICAR PG 2027 या फिर ICAR PG 2028 और एंटोमोलॉजी का माइंड मेकअप कर लिया है, तो आप हमारे ICAR JRF 2027 एंटोमोलॉजी के कोर्स में एनरोल हो सकते हैं, या फिर अगर आप 2028 में एनरोल कर सकते हैं अगर आप थर्ड ईयर के स्टूडेंट हो. अगर आपको इसमें कुछ भी क्यूरीज हैं आपकी, कौन कौन सी भी क्या क्या इसमें कवर कराया जाएगा, कौन कौन कवर कराया जाएगा, हर एक डिटेल आप हमारे टीम से बात कर सकते हो 9953077738 नंबर पे. इस कोर्स में आपको लाइव और रिकॉर्डेड क्लासेस मिलेगी, वीकली टेस्ट, यूनिट वाइज टेस्ट, फुल लेंथ मॉक टेस्ट, PDF नोट्स, वन-on-one मेंटरशिप, डाउट सेशंस, PYQ सेशंस, ईच एंड एवरीथिंग विल बी देयर. सो आप लोग आराम से इस वीडियो को देखें, अच्छे तरीके से आप इंसेक्ट फिजियोलॉजी पढ़ें. सो आई विल वेलकम रघु वंशी सर, जो आपको आज पढ़ाने वाले हैं इंसेक्ट फिजियोलॉजी. प्लीज सर प्लीज बच्चों को आज अच्छे तरीके से डेमो दे दीजिए कि कैसे IARI वाला पढ़ाता है. टॉपिक.
Hello my dear students. Myself I am D. Raghu and I have secured All India Rank 1 in ICAR JRF exam in the 2025, and I have completed my BSc from Professor Jaishankar, Telangana Agriculture University. This is a minute introduction of mine. So today I will start the demo class with insect physiology, and in that insect physiology the first system is the basic and the important one, that is the digestive system. Okay. So while coming to physiology, we all know that physiology includes all the internal anatomy and its physiological process. So in that physiology, we are having the different systems like digestive, respiratory, circulatory, excretory system, and muscular system and nervous system. In that, the basic system is digestive system. So here we can see the two words, that is V.B. Wigglesworth. So he is known as the father of insect physiology, and he worked on the Rodnius prolixus. So what is this Rodnius prolixus? It is a hemipteran bug. Okay, so normally it belongs to the family Reduidae, which is also called as corn nose bugs, assassin bugs and all. So, and why he selected this bug and what are the contributions made by Wigglesworth, we can see in the history portion there itself. Okay. So here in this class we will see the digestive system and digestion in insects, and mainly we will cover it in two aspects. One is structural organization and one is the complete digestion process. So normal organs of digestion, the digestive system includes the esophageal tube, means that alimentary canal from mouth to anus and its accessory glands. So combination of these two, and these accessory glands include salivary glands and gastric caecane. Okay, so combination of alimentary canal and these accessory glands completely is known as a digestive system. It's a basic. Okay, And while coming to the, we are having the different food habits even in insects or even in humans, like carnivorous insects and phytophagous and then all. So the length of this alimentary canal is longer in phytophagous insects when compared to the carnivorous insects. Why? Why length of alimentary canal is longer in the phytophagous insects? Phytophagous insects, the name itself tells it feeds on the plant. So plants have this cellulose content. So it will take more time for digestion and absorption. So when this compared, the portion of the midgut will be longer. So that's why the length of alimentary canal is longer in phytophagous insects than in carnivorous insects. Okay. And earliest stages of parasitic insects absorb nutrients through body surface. So earliest stages means the first instar larvae or second instar larvae will not have the completely developed digestive system. That's why they absorb the nutrients from the body surface itself. So this alimentary canal is divided into three parts mainly. One is foregut, midgut and hindgut. In this, the foregut and hindgut are ectodermal in origin and midgut is endodermal in origin. And foregut is called as stomatium, midgut is called as mesenteron or ventriculus and hindgut is called as a proctadium. And for this separation, in between we also have the two different valves. So we will see it in the upcoming slides. So alimentary canal is divided into three parts, that is foregut stomatium, which is ectodermal in origin, and midgut mesenteron or ventriculus, that is endodermal in origin, and hindgut or proctadium, it is also ectodermal in origin. Okay. So this is the complete idea of the digestive system. Okay. So here we can see the alimentary canal representation. So it starts with the mouth and ends with the anus, natural openings. After mouth we will get the pharynx, then after esophagus, crop and gizzard. Until the proventriculus or gizzard, it is the part of foregut. Okay. So until this portion, it is foregut. Then after we will have the midgut CK, ventriculus and the pyrolus site. This pyrolus site is important where the starting point of Malpighian tube. It is the entry site of Malpighian tubules. So until that portion it comes in the midgut. So next remaining portion is ileum, colon and rectum and anus. And this portion comes in the hindgut. This is the brief idea about the alimentary canal. In foregut it includes mouth, pharynx, esophagus, crop and gizzard. And in the midgut it includes midgut CK, ventriculus and filter chamber in case of homopteran insects. Okay, and pyrolus. And here to here we will have one wall, that is stomatial wall, and from midgut to hindgut we will have another wall, that is proctadial wall. Okay, which controls the reverse movement. So while coming to foregut, it is the anterior invagination of ectoderm. Okay, and this foregut will not be involved in any absorption or secretion process. Okay, and both foregut and hindgut are having the cuticular lining, that is intima. So here we can see there is a cuticular lining and intima, and during each molt it is also shedded and again reformed in the insect body. So that cuticular lining protects the foregut from any hard particles. Okay, so cuticular lining is unsclerotized and consists only in endocuticle and epicuticle. But in many insects, these are sclerotized and which is having the spines or tooth. So we will see that examples in upcoming slide. So here foregut again is divided into, for our convention, pharynx, esophagus, crop and gizzard. And gizzard is also called as a proventriculus. Okay, and here we can see the four parts. Then after, internally foregut layer consists of, it starts with the innermost intima layer, that is cuticular lining, then after epithelial cells, then after basement membrane and longitudinal muscles and circular muscles. Mostly these are the layers which is found in even midgut also. But there is change in the position. There first we will see the circular muscles, then after longitudinal muscles. But here in foregut we will see the order wise innermost intima layer, epithelial cells, basement membrane, longitudinal muscles and circular muscles. Okay. So in the previous slide we have seen that in many species there are sclerotized spines. So these spines helps in the grinding purpose, simply to tear any food substance. So here we can see the different direction of spines. So that's it. And esophagus, mouth and spines. Okay, and before that mouth, that terminal mouth parts led to the preoral cavity, and there is a epipharynx and hypopharynx parts will be there in that. So preoral cavity between epipharynx and hypopharynx is called as a cybarium, and preoral cavity between hypopharynx and salivary duct is called as salivarium. So these 2 points are major important, so we need to concentrate here. Preoral cavity between the epipharynx and hypopharynx is called as cybarium. Preoral cavity between hypopharynx and salivary duct is called as salivarium. Okay, so these are just introduction. Now we were jumping into the parts of foregut. So mouth completed, then after pharynx. So pharynx is a musculated organ. It acts as like a sucking pump. Because of this pump action, the sap feeders are able to intake the juice from the plants or the liquid content from the plants. So it is a musculated organ acting as a sucking pump in the sap feeders. So esophagus, it is poorly developed in a hemimetabolous insects, but in many adult holometabolous insects, it is a long slender tube running between the flight muscles and back to the abdomen. And esophagus of the insects feeding on highly resinous plants bears a single or paired diverticula. Diverticula is like simple pouch like a small structure that is formed by the esophagus. Mainly this modification we can see in case of the insects feeding on the highly resinous plants. So what this diverticula is used for? So insects which feed on this resin will store its resin in diverticula, which is modification of esophagus. So this is the complete structure. I have taken it from Insecta book, so it's easy to understand. So here we can see the preoral cavity, the cybarium between epipharynx and hypopharynx. Then after hypopharynx and salivary duct, we can see the salivarium. See, this portion is cybarium. This is here salivarium until salivary duct. Okay, these are the 2 parts. Then after mouth we will have then all mouth parts, then after pharynx, then esophagus, then here only we can see the salivary gland attachment, then foregut, crop and denticles. And here I said no, there will be 2 valves. First one is stomatial valve that controls the movement of food from midgut to foregut, reverse direction. Okay, but it allows the movement from foregut to midgut. Then after ventriculus, that is mesenteron or midgut. Then after at the site of pyrolus we are having the Malpighian tubules. Then after from midgut to hindgut we are having the another valve, that is the pyrolic valve. So this movement of the food is done by majorly 2 valves. One is stomatial valve and another one is pyrolic valve. So this is the second valve. Then in the rest hindgut portion we will have the ileum, colon and rectum and the anus. 2 natural openings. One is starting with mouth and ending with the anus. So in between we will find some layers such as intima, then after peritrophic membrane and all. So we will see in upcoming slides. So another thing, example caterpillars, and we have discussed there in the previous slide. Esophagus is modified into single or paired diverticula in the insects which feed on the highly resinous plants, and this diverticula is used to store that resin. So for that example is caterpillars of Myraska genus, commonly called as the concealer moths, feeds on the Myrtaceae family plants. And okay, so here it is the one statement. And sawfly larva in the genus Neoprodon which feeds on the pines. So in both the cases we can see this diverticula. So here, so mostly students what they will do, okay, caterpillars of Myraska genus is also known as concealer moths, will feed on the Myrtaceae family members. But what includes this Myrtaceae family members? So in that, what are economical or agriculturally important one? So mainly the first member is Gava and Jamuns we will get. and some other wood forest plants. So when compared with the fruit crops, the first plant we will get is guava. So it is also having the high resonant canal channels. Okay, so here one entry mouth will be there, esophagus will be there, pharynx will be there. Then after esophagus normally in the typical structure or the most representative structure we will draw it as a simple tube. But here in some insects there is a modification, it forms the diverticulum, whether it may be single or paired or anything. So in that diverticulum the resin is stored. Okay, and rest the crop, midgut and rest portion will be same. Okay, so we have completed the pharynx and esophagus. Then we are coming to the third part, that is crop. Crop is a dilated distal portion. Simply it acts as a food reservoir. So the food is taken through the mouth and it came to the pharynx, then traveled through the esophagus. Now it should be stored somewhere for further action. That storage conditions or the reservoiring conditions will be provided by crop. Okay, in adult, the Diptera and the Lepidoptera, it is a lateral diverticulum of the esophagus. And the walls of the crop are folded longitudinally and transversely, and the folds become flattened as the crop is filled, permitting a very large increase in air. So if this is the crop, the layers will be like this sometimes in different foldings, longitudinally or transversely. And when the food is taken, these layers will become flattened, so that it will helps in increasing the volume. If it is increasing the volume, it helps the conditions of storage. And in Periplaneta, that is cockroach. In Periplaneta, the crop does not contain food, but it is filled with air. In bees, the crop is called as a honey stomach, where nectar conversion occurs. And this is, we know. Which part of bees is called as a honey stomach? Why honey process or the nectar conversion happens in the crop portion? That's why the crop in the bee specifically is called as honey stomach. Then proventriculus or gizzard. Mainly the gizzard or proventriculus is found in solid feeders, and it was absent mostly, otherwise very much reduced in the sap feeders. And so why it goes like that? And proventriculus helps in the digesting the hard substances. So that's why it is useful in the solid feeders. In case of sap feeders or fluid feeders, just it will suck the sap from the plant, which is, it will not have any hard particles. So there is no use of this gizzard. That's why it is absent evolutionary changes, or it may be much reduced in the fluid feeders or the sap feeders. Okay, and proventriculus prevents the passage of solid food from midgut to foregut. It permits the movement of liquids in both directions. So here, this, one minute. Yeah, proventriculus prevents the passage of food from midgut to foregut, reverse direction. But it is the solid food, but it will permit the movement of liquids in both direction, whether it may be foregut to midgut or midgut to foregut. So the internal cuticle of the gizzard, we have said already, the entire foregut and hindgut is lined by the cuticular lining that is called as intima. So this internal cuticular layer is modified. So in different insects it is modified differently. So here we will look into these examples. In cockroaches, crickets and grasshoppers has 6 strong chitinous plates or teeth which grind and strain the food. So what are these? So in cockroaches, crickets and grasshoppers has 6 strong chitinous plates. So this cuticle is formed as a strong plates which helps in the grinding and staining the food, and plate like in honeybees to separate the pollen grains from the nectar. This is another modification. And spine-like in the flea to break the blood corpuscles, and we know these fleas will feed on the blood or the blood sucking insects. These are normally, so to break that corpuscles, whether it may be RBC or WBC, it is modified into a spine like, so to tear that cells. So spine like in flea. And food flow from foregut to midgut is regulated through, I have said no, it is called as cardial valve or esophageal valve or stomatal valve. All three names indicate the single valve. So here we can say cardial valve, esophageal valve and stomatal valve. So it regulates the movement of food from foregut to midgut. Yeah. With this we have completed the foregut portion. The foregut includes pharynx, esophagus, crop and gizzard. So this is the order which the insect is consisting. So in that proventriculus, it helps in the grinding of the solid food particles. So it is present in solid feeders and mostly absent, or it might be much reduced in case of the fluid feeders. Okay, and the food regulation is done by stomatal valve, which is also called as cardial valve or esophageal valve. All three names single and same. Okay. And while coming to the next portion, that is midgut, which is also called as mesentron or ventriculus. So gizzard is proventriculus before the midgut. So it is proventriculus and midgut is ventriculus. So here we cannot see any cuticular lining. No cuticular lining in the midgut. And most digestion and absorption takes place in the midgut itself. Why? Here there is no layer. So and many of the enzymes were released from this midgut portion, and this epithelial layer is having the different types of cells which is having the different functions. Okay, and pH of the midgut is 6 to 8 and it's important, pH. Structurally midgut consists of, there also we have discussed, internally the foregut layer consists of intima layer, basement membrane, epithelial cells, then after longitudinal muscles and circular muscles. Here also the same layers will be there, but here in case of intima we are having the peritrophic membrane. And epithelial cells, that is AC, BM is basement membrane, CM is circular muscles, LM is longitudinal muscles. So there, what is the difference between those layered structures? There circular muscles was followed by longitudinal muscles, but here in case of midgut arrangement, circular muscles come first and then after longitudinal muscles. So epithelial layer of midgut consists of 3 types of cells, and it's very much important from this portion. Okay. So midgut epithelial cells are mainly 3. One is secretory cells. So these secretory cells is also called as enterocytes or the principal cells. What is this principal cells? So it is having the major action or the activity. So what the activity? Mainly it releases the enzymes. Okay, so it is the important, so it's a principal cells. Then after columnar in shape, that's why they are also called as columnar cells. Secretory cells, enterocytes, principal cells or columnary cells. This releases enzymes through the microvilli arranged in a brush border or honeycomb border. It's an arrangement, but you know what is microvilli? And the difference between villi and microvilli is also important. So here the secretory cells or enterocytes or principal cells or columnar cells. So why secretory cells? They are secreting enzymes. Principal cells, it is the cells which is having the major function, and columnar cells, the shape of the cells is columnar, hence the another name is columnar cells. So all this major function is releases enzyme through microvilli. So next is goblet cells. So what are these goblet cells? Aged secretory cells is known as a goblet cells. Here we can read. Aged secretory cells is known as goblet cells and mainly serve for storage, one function, excretion, another function, and mediate the alkalization of the midgut. We know the pH scale. If 1 to 6 it's acidic and exact the 7 is neutral, and after that it's alkaline. So maintaining this alkalization, mediate the alkalization of the midgut is the function done by goblet cells. And how it is done? Via K plus or 2H plus antipode that is driven by electrogenic 5 ATPase. We will see in upcoming slide this image. So the function of goblet cells is storage, excretion, and mediate the alkalization of the midgut to maintain that particular specific pH, that is 6 to 8. Okay, so 2 cells we have completed. Then the third one is regenerative cells, and while coming here, the goblet cells in Lepidoptera is flask shaped when compared. So here we can see flask shaped goblet cell, opening of the midgut, then after cavity, in that again the layers mitochondria, nucleus and basilar laminar membrane, and the rest of the hemosyl fluid will be there. So it is in the flask shape in case of Lepidoptera, and KH plus and 2H plus antipodal we can see. Yeah. And third type of cell is regenerative cells or stem cells. We know regenerative or stem cells replaces the secretory cells. Okay, regenerative cells or stem cells replaces secretory cells by the formation and production of new cells. So we are having totally 3 types of cells. One is secretory cells, which is also called as principal cells or enterocytes. Then after columnar cells, and that aged secretory cells is called as a goblet cells, which is used for storage, excretion and alkalization of the midgut. And here the third type of cell is regenerative cell or the stem cell. Stem cell replaces secretory cells and by formation and production of new cells. And regenerative cells may be arranged in groups or clusters or remain scattered singly, whether they may be in single cell or maybe they may be arranged in groups or clusters. If they are arranged in groups or clusters, they are known as nidhi. The most important, they are known as nidhi. Why nidhi? Nidhi is somewhat precious. The name itself, the technical meaning of nidhi is which is precious and which is valuable. So this regenerative cells or stem cells replaces the secretory cells. So what happened after formation of secretory cells? If they are aged, they are converted into goblet cells. Then after some days the cell function will be stopped. Then after such cells are usually replaced by the regenerative cells. So their main function is again becomes to release the enzymes. So that is why they are precious or valuable. The regenerative cells which may be arranged in groups or some small clusters, they are called as nidhi. Otherwise they may be also in single. Okay. In some beetles, the nidhi are restricted to crypts, which are visible as small papillae on the outside of the midgut. Here also we can see the image. So actually, what nidhi exactly? This is the gut lumen. This is the villi, microvilli layer. Then after, here we can see the enterocyte. Fully developed enterocyte. That enterocyte is secretory cell or principal cell or columnar cell. Then after differentiating cell, we can see the differentiating cell. Then after we can see the stem cell, and this arrangement of stem cells in group is called as nidhi. Okay. So in case of some beetles, this nidhi forms a small papillae. So this is the structure of papillae. Here we can see again gut lumen, the crypt, the crescent. Then after here we can see the rest of that extended structure or outward structure, that is known as papillae. In that papillae we can see the regenerative cells, and longitudinal muscles, circular muscles are all of same for lower layers. Yes. So important, the epithelial layer cells are of 3 types. First one is secretory, second one is goblet cells, and third one is regenerative cells. Okay. So important structures in midgut. So in foregut also we have divided into 4. Again in midgut also we will divide it into 4. One is peritrophic membrane and gastric CK and filter chamber in homopteran insects specifically. Otherwise there will be 3. And fourth one is pyrolic valve. So gastric CK is also known as enteric CK or hepatic CK. All the 3 names are same: gastric CK, enteric CK and hepatic CK. Mostly these are 2 to 8 in number at the proximal end of the midgut. Okay, and sometimes at the posterior end also we can see. So in that case, in which insects we can see? Mainly the coleoptera and heteroptera insects. We can see this gastric CK. So proximal means first. So that portion we have seen in the typical representation. So sometimes it may be in the posterior end also. In that case specifically we can observe in the coleoptera and heteroptera insects. So this is the range. So this is the range, 2 to 8 in number, but we will have some more modifications in that in between range. So we will see, for example, the crickets that belong to the family Grylidae and some diptera and larvae will have 2 CK in number, and grasshoppers or that belongs to the family Acrididae will have 6 CK. Here you can see. In crickets and some diptera and larvae there are only 2 CK, and in grasshoppers that belongs to the family Acrididae are having the 6 CK. Cockroaches and larval Culicidae, this Culicidae family belongs to the diptera, and mantids are having the 8 CK. See, 2 to 8. So it is the range of the gastric CK or enteric or hepatic CK. In case of crickets and some diptera and larvae we can see the 2 CK, and in grasshoppers we can see the 6, and in cockroaches and larval Culicidae we can see the 8 CK. So what is the structure? This structure increases the functional area of midgut. So if there is a lot of absorbing, if function area is there is a lot of absorption will takes place, so that all the nutrients from the food can be absorbed. So this structure increases the functional area of the midgut. And in Columbola, that is springtails, and Lepidoptera, the moths and butterflies, these are generally wanting. It means they are absent in these 2 orders, springtails and moths and butterflies, that is Columbola and Lepidoptera. These are generally wanting, means they are absent. So normal range is 2 to 8. In different insects we can observe the different number of CK. So first one is gastric CK, then after coming to peritrophic membrane. So this, some books, if you read different books, the peritrophic membrane is also called as peritrophic envelope or peritrophic matrix. All are same. Okay, the other names. So it is the internal lining of the midgut, secreted by the midgut epithelial cells only. So it is also present in solid feeders and absent in sap feeders. So mostly this is the thing students will read. Present in solid feeders, it is absent in sap feeders. Then we need to know what sap feeders is having or why it is absent in sap feeders. Only this kind of reading will help you in understanding the concept or the subject, so that you can answer any questions. So present in solid feeders and absent in sap feeders such as Hemiptera, Lepidoptera, Thysanoptera and endoparasitic insects, that is Strepsiptera. Okay, and it lubricates, facilitates food movement and protect the epithelial cells from the harder food particles. So here we can see who will take harder food particles. Definitely solid feeders will take. That's why it is present in solid feeders. But in case of fluid feeders or sap feeders, they will take only liquid. There will not be any harder food particles. So it is absent in that. But in place of peritrophic membrane, there is another lipoprotein layer. That layer is called as perimicrovillar membrane, forms complex folds. In usual books, what students prefer for JRF books, you will not find this point in any book. Okay, so it's important. In place of peritrophic membrane, in case of sap feeders, there is one more lipoprotein layer, that is perimicrovillar membrane, which forms the complex folds. Why perimicrovillar membrane? So microvilli means it will have the larger functional area. So it will releases enzymes, then after it will helps in absorbing the all the nutrients from the sap. So it is the function it does. So here there is no what chance of damaging the peritrophic perimicrovillar membrane because of absence of harder food particles. So this peritrophic membrane is present in case of solid feeders and absent in case of sap feeders. This is thing. And the third one is pyrolic or proctadial valve. We have discussed from foregut to midgut, the movement of food is regulated by the stomedial valve, which is also called as esophageal or cardial valve. And in case of from midgut to hindgut, this valve regulates it. The midgut opens into hindgut through a pyrolic valve which regulates food flow. In some immature stages of insect, midgut is not connected to hindgut till pupation. That is most known example is honeybee grub. Okay. So two valves are important. Then after the fourth part, it is not present in all insects. Mainly it is present in the homopteran insects. Filter chamber. What is this filter chamber? Simple. Anterior part of midgut will bound to the anterior part of the hindgut and it forms the one chamber which filters the food. The name itself tells it is the filter chamber. But with this structure or the modification, what is the benefit that insect is having? That is important. So this filter chamber enables the excess fluids, including the sugar in the food, to pass directly from the anterior part of the midgut to the hindgut without passing through the middle portion of the midgut, and thus preventing the excessive dilution of hemolymph enzymes and facilitate better enzyme activity. So midgut is helps in the filtration or absorption. So if that goes, there will be excessive dilution process. So this filter chamber reduces the length and simply it will passes through it. So all the particles will be filtered out. So it helps in the excessive dilution, okay, of the enzymes. Then after it will increases the better enzymatic activity. Here we are having three important functions. One is enables excess fluids including the sugar in the food to pass directly from anterior part of midgut to hindgut without passing through the middle portion of midgut, thus preventing the excessive dilution of hemolymph is one function. And dilution of the enzymes. If there is no dilution of enzymes, definitely enzyme activity will be better. So facilitate the better enzymatic activity. What happens if enzyme is diluted? Enzyme won't work properly. It will not do its work. So it will prevent the excessive dilution of hemolymph and enzymes. So gradually or obviously the better we can say the better enzymatic activity. So we will have the image here. Simple filter chamber. See here we can see the esophagus, then anterior midgut is attached to the anterior hindgut. Okay, then after in between we can see the Malpighian tubules and all. So this modification is specified in homopteran insects. Then rest of the parts will be same: ileum, colon, rectum and posterior midgut. This will be same. So we have completed midgut also. In midgut also again 4 parts are there. One is midgut CK, that is enteric or hepatic CK. Then after pyrolic valve, then filter chamber and all. Yeah. And in the coming to hindgut, that is proctadium, which is also a ectodermal in origin. The layer is same, ectoderm. If it is anterior invagination, it forms the first, that is foregut, and posterior invagination is hindgut or proctadium. Hindgut is also having the cuticular lining. It is known as also intima, same as the foregut. But what is the difference between these two intimas? So when compared to the foregut, the hindgut intima is very thin and more permeable. It helps in absorption. Simply it helps in absorption. There in foregut intima we cannot see the any secretion or absorption process. But here we can see little bit absorption, not in much quantities but in minor quantities. Why? Because intima layer is thin. So it is the difference between those foregut and hindgut intima. The anterior part of hindgut is called as pyrolus, is the site of entry of the Malpighian tubules and possessing the pyrolytic valve. So I said earlier in the picture itself, the site of entry of Malpighian tubules is marked by the pyrolus, and there itself we can see the pyrolytic valve that regulates the movement of food from midgut to hindgut, and from foregut to midgut it is controlled by the stomatial valve. Okay, and here also hindgut is divided into three parts: ileum, colon and rectum. Three parts. So here Malpighian tubules, regulation of salt, water and nitrogenous waste excretion. These are the main functions done by Malpighian tubules. Apart from that, there we need to learn the what is the Malpighian tubule, who gave it, what is its structure, and what are the different modifications in case of Malpighian tubules and different functions apart from this. So this all portion we will learn it in the excretory system. But until here, just for your understanding, Malpighian tubules helps in the regulation of salt, water and nitrogenous waste excretion. So hindgut is divided into ileum, colon and rectum. So first one is ileum, which is also called as intestinum. The narrow tube between the pyrolus and the rectum, mostly single cell type is present. So here we can see. So this is the gut lumen. In the hindgut also we will have the cuticle lining, that is intima. So here the folds of apical plasma membrane we can see. Then septate junction, basal lamina. Then after here folds of basal plasma membrane. So why here also there is a difference. By the image itself we can see it. So apical plasma membrane, it's somewhat freely folded, and we can see the wider space between them and the number of mitochondria. So these are the mitochondria, here we can see like that, mitochondria, and this mitochondria count or the number of mitochondria is less in compared to folds of apical plasma membrane. So here I will write okay. And in case of folds of basal plasma membrane, it is densely folded. See here, many foldings were there, and the number of mitochondria is also more in case of basal plasma membrane. Okay. So this is the cell of the ileum, that is a mostly single type of cell. We can see the folds of apical plasma membrane and folds of basal plasma membrane. So what is the difference between these two folds is that is loosely folded or somewhat wider space is there, and the count of mitochondria is less in apical plasma membrane when compared to the basal plasma membrane. And ileum is expanded to house the microbes, forming the pouch or pouch in termites. In many books you will find a pouch or pouch, both are same in termites. In case of termites, this ileum helps in forming the one storage house of the microbes. That storage house is known as pouch or pouch in case of termites. And fermentation chamber in larval scarabidae. The scarabidae includes chafer and dung beetles. So here we are finding the word scarabidae, that is one of the family of Coleoptera. So if we see such families, again we need to interlink with this with taxonomy. So how? What are the different characters of scarabidae? What are its example? If scarabidae is there, some of the modifications, fossorial legs will be there. Then after, extensive parental care will be there. So these are the two important points when coming to the scarabidae. So when we read anything, we need to interlink with each other so that we can remember it better, and our understanding and clarity of that particular concept will be more. Okay, so all the time I will suggest students to interlink the points. Okay, so ileum is expanded to house the microbes and forming the pouch or pouch in termites, and fermentation chamber in larval scarabidae. Okay, so here we can see the diagrams. So first one is scarab larva, the scarabidae. So this ileum forms the fermentation chamber. Here we can see this fermentation chamber. So what this fermentation chamber will do? Actually, what this Carabidae will feed? The soil particles. It will be laid in the soil and it will feed on the available matter in the soil. Then after the termites, the pounds, which acts as a house of microbes. So it is having the gut symbiosis in termites. Okay. Some higher termites will have something and lower termites will have something, and we will see in case of termites. And colon, the distal part of ileum is colon. Simple. The distal part of ileum is the last portion of the ileum is known as colon. And the next one is rectum. Rectum is a thin wall chamber again. It is also having the pads. That pads is known as muscular rectal pads, or simply rectal pads. Rectal pads are usually 6 in number. Rectal pads are usually 6 in number, arranged radially, and the rectum of the may be papillae-form in case of Diptera. So rectal pads play a role in the water absorption and essential ions and amino acids from the urine. So what is the function done by these rectal pads? Rectal pads play a role in the water absorption and absorption of essential ions and amino acids. The rectum is the site of respiratory gas exchange in some aquatic insects, that is the larvae of Anisotera. So Odonata is divided into Isotera and Anisotera. In that, specifically the site of respiratory gas exchange can be seen in the aquatic forms, that is dragonflies, in which the proximal rectum is modified to form a branchial chamber. It is important. Sometimes the question may come, in which of the following insects the rectum is modified into branchial chamber? They will give some flies, fruit fly and another fly, then after the dragonfly, damselfly. But this modification we can see in the dragonfly. That this modification helps as a site of respiratory gas exchange for respiration. So we have said the rectal pads. So here we can see the rectum cross section. In that, here we can see easily these folds kinds of thing. These are the rectal pads. Okay. Then after section through a rectal pad, so here we can see the individual rectal pad again. So cuticle will be there, septate junction, principal cells, calariform junction, basal, trachea and hemocoel. So in case of dragonflies, we said it acts as a site of respiratory gas exchange. It's nothing but it acting as a simple respiratory system. Why? Because this rectum is modified into branchial chamber. So it need to have the trachea to carry the oxygen. So that trachea we can see here. See the tracheal. Here we can see the tracheal extensions. So this will carry the oxygen to it. Trachea and tracheals. With this we have completed the structural organization. Only the part of structural organization. We said digestive system consists of alimentary canal and its accessory glands, mainly salivary glands. So here the first gland is salivary gland. In cockroach, a pair of labial glands acts as a salivary glands, where salivary ducts are open into salivarium. And in case of caterpillars, the mandibular glands secrete saliva. And in silkworms, the salivary glands forms into silk glands which secrete the silk. And these silk consist of fibroin and sericin, and they will be modified into brave and all, which changes into elastic protein, fibroin and sericin. So what are the different functions of saliva? In cockroach, the saliva contains amylase. So what this amylase does? It does the digestion of starch or breakdown of starch, the major enzyme to break the digestion. So amylase, digestion of starch, is the important function of saliva in case of cockroach. And in honeybee, we can see the enzyme invertase, which helps in the digestion of sucrose. And in plant bug, pectinase stylet penetration and extra intestinal digestion, it will help. So the middle lamellae of the plant cells will have the pectates. So these plant bugs will have the pectinase to dissolve that pectin. If it is dissolved, can be easily penetrated stylet. So which helps in the extra intestinal digestion. Then after in mosquito, we can see a anticoagulant here, and that prevents the blood clotting. That's why when the mosquito bites, after completion of its feeding, when it takes out its stylet back, at that time only we will feel the pain. Why? Because until then it will release the anticoagulant, it will prevent the blood clotting. So after that, what are their remaining functions? In gall producing midges, the saliva contains the indoleacetic acid. What this indoleacetic acid does? It will help in hypertrophy and hyperplasia. One is cell increase in size and the increase in count. So this indoleacetic acid production helps in gall producing. And in gessetes, the saliva contains the lipase for digestion of lipids simply, and protease for the protein digestion, and it contains the toxin which produces the phytotoxemia condition. Is nothing but while feeding it will release the toxin. After that the plant will produce as the tissue necrosis. It's nothing but drying of tissues and gradually killing of the plant. So this condition is called as a phytotoxemia. Okay, and the next important digestive gland is hepatic CK or midgut epithelial cells. We have already seen in secretes most of the digestive juices, and here two types of cells were involved in the enzyme secretion, that is holocrine and merocrine cells will be there. So here I can draw a diagram for you. So holocrine and merocrine cells, the cells will be same, but in case of holocrine cells, when they want to release the enzyme or any other product, any metabolite, only the epithelial cells will disintegrate, and so these cells will go. And in merocrine, the enzyme secretion occurs without any cell break. Just normally they will release enzymes, but there is no disintegration of cells. That is the major difference between the holocrine and merocrine. Holocrine cells, the complete cell will disintegrate. Merocrine cell, without any disintegration or without any cell break, the enzyme secretion occurs. Only that is the difference. And microbes in digestion. In insects, cells are housing symbiotic microorganisms. That cell is called as a mycetocyte. Important. These mycetocytes aggregate to form an organ called as a mycetoma. Okay, mycetocyte is individual cells. Mycetoma is group of cells. Simple. This group of cells will form tissues, and tissues will forming the organ. Okay, so that organ is called as mycetoma, and individual housing cell is called as mycetocytes. In case of flagellate protozoa, it produces cellulase for cellulose digestion. In case of termites and wood feeding cockroach, Cryptoceras species is there in wood feeding cockroach. So it is the close relative of the termites, Cryptoceras. So this all we will read it in taxonomy. And flagellate protozoa, it produces cellulase for cellulose digestion in case of termites and wood cockroach. So previously what they used to tell me is these termites will fall under the Isoptera, but now the Isoptera became the infraorder. Now they were a separate order known as Blattioidea that only consists of the termites. So this classification, when we go into this taxonomy and history portion, there we can see. And in bacteria, it helps in the wax digestion in case of wax moth. This wax moth may be greater wax moth or lesser wax moth. In case of pest of honeybees, we will read it. Galeria melanonella, Acrocia grisae will be there. Two species, one is greater wax moth and lesser wax moth, and this bacteria will help in the wax digestion. And bedbug and cockroach would obtain vitamin and amino acids from microbes. These microbes were transmitted between the individuals through food, that is mouth to mouth feeding, that is called as a trophallaxis. We know the sharing of the food between the insects from mouth to mouth is called as trophallaxis, and if they are transferred to eggs to the next generation through the egg, it is called as a transovarial transmission. So both are important. So here we can see the digestive enzymes, the insect group, enzymes and substrate. So phytophagous insect that feeds on the plant, amylase, maltase, invertase are required, omnivorous protease, lipase, and meat eating maggots that requires the collagenase. Why? Because in meat we will have the muscle protein, collagen will be there. To digest that collagen, we require the collagenase. So in bird lice we can see the keratinase, hair protein. So this is until the structural organization, the both alimentary canal and its digestive organs, the glands, salivary glands and gastric cache. Now we were coming into the process of digestion. Simple: ingestion, transportation, digestion, absorption and digestion. So ingestion, it will intake the food. Okay, that is known as ingestion. Transportation, by peristatic movements of those layers it will move. And digestion, it some this we have seen that secretory cells of the midgut will produce enzymes. And absorption we will see, and digestion through anus it will come. And ingestion, before ingestion the food is somewhat digested before the actual intake, that is called as extraintestinal digestion. In case of plant bugs we have seen it. So this is the series of steps: ingestion, transportation, digestion, absorption and digestion. So that is the process of digestion. Apart from that, we need to know somewhat. All the time we cannot expect the question, but we need to be ready for whatever the question came. At least we need to have. Apart from considering the beside exam or something, we were the students of entomology and we will continue in this field. So we need to have that broad knowledge related to digestive system, and here somewhat points I have added and we will look into it. Insect diet with mainly diet are mainly of three types, that is holotic diet, meridic diet and algodic diet. And holotic diet means all the components in diet are known to us. Okay, and mainly it is consists of the pure chemicals. Holotic diet consists of pure chemicals without natural food material, exclusively made of purified chemicals, and complete composition is known to us. So where this condition we can see holotic diet? In labs. The labs, if we are raising Spodoptera, Helicoverpa or any other insect, all the diet will be prepared by us and we know all the composition of it. That diet becomes the holotic diet. And meridic diet, both natural food materials and chemicals are combined in proper portion and prepared. Holotic diet is crude and extract of natural food material and most of the compounds being chemically unknown. This is used for mass rearing. And rearing of one or more individual of same species in non-living medium is exenic, and rearing of organism in association with one or more unknown species is known as xenic. See, two confusing statements. Rearing of one or more individuals with the same species is xenic, and rearing of organisms in associated with one or more unknown species is xenic. Stable protein in wool, hairs and feathers is keratin. And polypeptide chains, why it is stable, this keratin? Why? Because it is having the disulfide bonds in the keratin protein. This disulfide bonds will gives it the stability. Okay, and that's why in bird lice we will see the keratinase to digest it. And all insects require ascorbic acid in foodstuff mainly present in the plants and required for cuticular sclerotization. In case of this cuticle after structure and how the cuticular tanning process is going on and how this cuticle is formed, what are the biochemical pathway, we will get to know how the ascorbic acid is useful in it. So all insects require ascorbic acid for cuticular sclerotization. So now we will summarize the class little bit in case of flowchart. So my ways, if you draw any flowcharts or any mnemonics, it is easy to understanding and easy to remember also. So digestive system is there. One is structural organization, then after another one is accessory glands we are having. And here we have alimentary canal. So again this alimentary canal is divided into three different parts we have seen already. foregut, midgut and hindgut. And here again foregut is divided into pharynx, esophagus, then crop, then proventriculus or gizzard, 4 parts. Gizzard. And midgut is divided into midgut CK, then after peritrophic membrane, pyrolytic valve will be there, then after filter chamber. And hindgut, ileum, colon and rectum. So these are the different structural organization of alimentary canal we have seen. In case of glands, we have seen the salivary glands, labial glands, mandibular glands and everything, one thing. Then we have seen CK and microbes associated with it. So if we, any concept or any other subject, if we read it in such a flowchart manner, it helps us to easily understand. So if you could remember this flowchart, now you will get the entire idea. The alimentary canal is divided into foregut, midgut, hindgut, and foregut and hindgut are ectodermal in origin, midgut is endodermal in origin, and in foregut again we are having 4 parts: pharynx, esophagus, crop, proventriculus, and it is also called as gizzard. And while coming to midgut, it is divided into the peritrophic membrane and midgut CK, pyrolytic valve and filter chamber in case of homopteran insects, not in all the insects. And the hindgut, it is divided into ileum, colon and rectum. So now you can write easily one point under each thing. Pharynx, musculator organ, acts as sucking pump, esophagus, narrow tube, and in some cases like the insects which feed on high resinous plants, we can see the diverticulum. And crop, in honeybees it acts as a honey stomach and all. So like this you need to develop one flowcharts or mnemonics or simple diagrams according to yourself, so it helps in easy understanding. So I hope you liked the session. So currently we were running the 2 batches for it. One is for upcoming exam, that is ICAR JRF 2027, and another batch is ICAR JRF 2028. So we named it as a foundation batch. So that's it. So you will get the unit test after each lesson, that is weekly test. Then after you will get the analysis of the test, then after monthly and all the mock test and available notes.
All right then students, hope you all liked the class. कैसा लगा आप फीडबैक कमेंट सेक्शन में ज़रूर बताएं, जैसा भी लगा हो. और अगर आप, जैसे सर ने बताया कि आप कोर्सेज के बारे में देख सकते हैं, अगर आपको कोई भी इंक्वायरी करनी है नंबर हैं आप देख सकते हैं. विद दिस थैंक यू सो मच, टेक केयर ऑफ़ योरसेल्फ और अच्छे तरीके से तैयारी करो. जहाँ से भी करो बहुत अच्छे से करो. अगर आपको हमारी नीड लगती है कि आपको हम अच्छा पढ़ा सकते हैं, देन जॉइन IARI वाला. थैंक यू सो मच. ऑलराइट सर, थैंक यू सो मच.